Okay, in this little video I want to introduce you or refresh your memory about gene expression. So this is really a very short and quick version of how we get from a gene to a protein in a process that is known as transcription and translation. So what is our DNA? That's the first thing we have to look at. Well, the DNA stores important information for living organisms, and as we all know already, although from this class, life and function of cells depends critically on a large number of very different and very specific proteins. So for cells to function properly, they need enzymes to do a lot of intracellular work, but also enzymes are required and released by cells for important processes like, for example, digestion. Then, of course, in the membrane embedded, we have ion channels that allow the transmembrane transport of ions, very important for the electric signaling in neurons. Then we have other transporter proteins that are embedded into the cell membrane to allow exchange of other solute molecules across the cell membrane, either by active or passive transport. And then, of course, we have structural proteins, cytoskeletal elements that allow cells to maintain shape, to maintain their form, and also provide scaffolding for a number of biochemical processes within the cell. The problem with those proteins is that they are very easily disturbed, changed, and to the point that they become dysfunctional. This can happen by even minor changes in the environment. That's actually something that is sometimes important for their particular function. We'll find out more about that when we talk about how ion channels, for example, are gated, open and closed, because here changes in the protein structure are the key how this works. Much more dramatic are errors that happen during the synthesis of proteins, and this would be an error in the primary structure of a protein, its amino acid sequence. If anything goes wrong here in those circumstances, we may end up with proteins that do not function the way they're supposed to function. So what life needed and evolved over millions and millions of years is a system that makes sure that proteins are synthesized, built exactly in the right way by following a blueprint. And by doing that, by following that blueprint, a lot of the errors can be avoided that otherwise would lead to a change in protein structure that renders those proteins dysfunctional. And the other benefit of having this blueprint stored in cells is that it allows also the information, the code for producing proteins in the proper way can be passed on from one generation to the next generation. So, and this biological blueprint, the construction plan for cells and their proteins, is stored in the DNA of each and every cell. The RNA comes into play a little later when we look into how the information stored in the DNA is put to work during gene expression, because just like in the real world, well, not the real world, like the technical world out there, you really don't want to use your original copy of a construction plan for work. And we all do that, of course, although when we work in labs, we're not taking the textbook to the lab. We may just photocopy the pages that we need during the lab so that we, if something goes wrong, do not damage our original. So DNA, the genetic information that is stored in cells, is the abbreviation for deoxyribonucleic acid. And this is our blueprint, and the DNA is always present in cells and only in the form of a double-stranded molecule. The RNA, abbreviation for ribonucleic acid, is on the other hand not only our working copy of the DNA that we want to put to work, it's also, contrary to the DNA, always and only present in cells as a single-strand molecule. So what they both have in common is that both DNA and RNA are polymers formed by nucleotides. Nucleotides are a class of molecules found in cells that all have more or less the same basic construction. What they all have in common is a central sugar molecule, a pentose, a C5 sugar. The only difference here between our DNA and RNA is the type of sugar. It's in both cases a pentose sugar. In one case of the RNA, we have an OH group attached to one of the carbons of the sugar molecule, and in the deoxyribose that would be just a hydrogen ion. So that's the structural difference between RNA and DNA in what we call the universal backbone of each and every nucleotide that is used to assemble those polymers. And the other group that they all have in common is a phosphate group, shown here. And the third part in a nucleotide molecule is an organic nitrogenous base that is also attached to a carbon atom of the sugar molecule. And this organic nitrogenous base is probably the most variable part in those nucleotides used to assemble DNA and RNA polymers, because here we have a total of 5 different of those base molecules that are used for the different nucleotides. So here we have a type of nucleotides known as adenine, where this group is attached to the pentose. Then we have guanine, where a slightly different base is attached to the nucleotide backbone. Then we have cytosine, which is the third type of nucleotide that we find in the DNA. And the fourth one that we find in the DNA is thymine. Those 4 different nucleotides are the language used in encoding information in the DNA. In the RNA, we also use adenine, we use guanine, we use cytosine. And what's different between the DNA and RNA is that thymine of the DNA code is replaced by uracil in the RNA code. So each one, RNA and DNA, use 4 different nucleotides to encode information. Difference between RNA and DNA is thymine on the DNA side and uracil on the RNA side. So here we get now the next little detail that is important to know, that other than DNA only exists as a double-stranded molecule, also known as a double helix, is that we have here 2 DNA strands that are complementary to each other. In other words, they match. And they match in terms of how those nucleotide bases pair up in those 2 strands, because here thymine and adenine are complementary to each other, so they form pair, and cytosine and guanine are also complementary to each other. They form the other possible pairing. Okay, so here in the double strand of a DNA molecule, we always have complementary DNA base pairs facing each other in those two complementary DNA strands. In RNA, we also have this complementary pairing, in this case, of course, between uracil and adenine and cytosine and guanine. So it's important also in RNA that this base pairing happens, even if RNA is always only present as a single strand molecule. We'll see in a moment how that is important when it comes to the synthesis of proteins from individual amino acids. That's where we need also in the RNA world this compatible base pairing. So then, of course, we have to join those individual nucleotides into those DNA or RNA strands, and that happens by forming a chemical bond between two nucleotides. So in other words, here we remove a water molecule in the end by forming this nucleotide bond here between the phosphate group of one nucleotide and one of the sugar carbon atoms in the pentose sugar. This is called a phosphodiester bond for those of you that are interested in chemistry. And so here, by repeating that bond now here at the hydroxy group of our pentose here with the another phosphate group of another nucleotide, we can form this very long polymer molecule that forms our DNA or RNA strand. So now let's have a closer look how genetic information is encoded in the DNA. And the key here is that information is written into the blueprint of a gene in form of the nucleotide sequence. So in other words, what contains information is the sequence of different nucleotides in the DNA strand. So this A, T, C, C, G, G, T, and so on, that you may have seen when genes are sequenced in genetics or molecular biology. So we have those two complementary double-strand DNA molecules, two strands of DNA connected to each other, like this is shown here in this little diagram. And one of the two strands is always referred to as the coding strand. This is the one where the gene, the genetic information, is actually encoded in the nucleotide sequence of that DNA strand. And the other one is called the template strand. It's called template strand because it's used as the template to synthesize an RNA copy of the coding strand, because we have here those complementary bases in the nucleotides that will pair up accordingly. And by doing that, when we start out with the template strand, what we get in the next step is, we'll see that in a moment, an RNA strand that is the exact copy of the information that is contained in the DNA coding strand. So what's the trick or the sequence or the importance of those nucleotides in sequence here is how they are organized in triplets. The triplet is also known as a codon, and here a group of 3 nucleotides, we call that group of 3 a codon, stands for one particular amino acid in the amino acid sequence of the protein that is encoded in that gene, in that sequence of DNA. So here we see a table that shows us codons, and obviously, as you can tell here, since we see uracil in this table here, we're looking at the RNA molecule, the messenger RNA, mRNA, that is produced out of the template strand of the DNA, which is a perfect match of the coding strand of the DNA. And here we see that groups of 3 nucleotides encode for one particular amino acid. So this here is the genetic code that allows to encode information, and information here is the order of amino acids in a protein in both DNA and of course also in the RNA, which is the working copy that is then used to actually synthesize the protein from that DNA blueprint. So now of course errors can happen because a codon like this one here stands for one particular amino acid. Now if I change the reading frame, the way I read the sequence of nucleotides during the gene expression, I end up with reading the nucleotides in the wrong order, and then of course I assemble a protein from the wrong amino acids, which of course will result in something that will not work. So in a way, DNA is a molecule, and RNA of course as well, where we do not have any spacing between words, and a word here would be the instruction what amino acid to use, or codon here. So it's critically important that when we start reading our nucleotide sequence during gene expression, that we read it in the correct order, otherwise we only get nonsense as a result. And there are several mechanisms in place to make sure that during gene expression the DNA and of course later also the RNA is read in the correct and only one correct sequence of nucleotides. So the first step that happens during gene expression, the way from gene to protein, is what we refer to as transcription. So during transcription we use the template strand of the DNA double strand molecule to produce a complementary copy of mRNA. So you can see here G matches up with C, A matches up with T, T with A, C with G, etc., etc. Of course, now you probably already noticed that there's a little error in the diagram. So this T here, because we're looking at a messenger RNA molecule, would actually have to be a uracil. So here you can tell even textbooks sometimes have errors in it. So the process of generating a messenger RNA molecule from the DNA template strand is what we call transcription. So here, step by step, what happens? So here we have our two complementary DNA strands, the coding strand on the top, and we see here that the template strand is complementary to it in terms of base pairing of the nucleotides. Okay, so now we use the DNA template strand to transcribe it into the messenger RNA strand, which then of course results as an exact copy of the DNA coding strand. So this is the process here, transcription of the template DNA strand into a messenger molecule, a messenger RNA molecule. And here again we have now that complementary base pairing between the T and A, the A and U here because T in the messenger RNA is replaced by U in the the T in the template strand is replaced by a U in the, or paired up with the uracil in the messenger RNA. And if we replace here, just for clarity reasons, the U's with T's, what we see then is that the messenger RNA that was generated as a complementary copy of the template strand is an exact match of the coding strand of the DNA. So we transcribed our DNA coding DNA strand by using the template DNA strand into a messenger RNA molecule. And this process is called transcription because we still keep information in the language of genes in the nucleotide language. We just transcribe it into, well, let's say, different dialect by replacing thymine in the DNA code with uracil in the messenger RNA coding. So now we're good to go with step number 2, which is known as translation, where we use the messenger RNA molecule that was prepared in the previous step, in the transcription step, to synthesize a protein. So here now we take the messenger RNA as the working copy of the DNA coding strand to produce the protein that is encoded in that gene on the coding strand of the DNA. This is a process called translation, because now we have to translate the nucleotide language of RNA and DNA into the amino acid language of proteins. Because just like the nucleotide sequence in both DNA and mRNA contains information in terms of what the order of amino acids in the encoded protein is, we have to translate that from nucleotides into proteins. That's why this step is called translation. And all this happens in the cytoplasm of the cell, where the machinery for translation is located. To do the job, we need, of course, our working copy of the gene, the messenger RNA. Then we find another type of RNA that is known as tRNA, or transfer RNA. An example is shown right here in the diagram. So it has this weird cross-shaped appearance, and there are two parts that are important. We have here the anticodon loop, which is a complementary base pair match to the codon on the messenger RNA. So here we have a whole number of different tRNA molecules that are able to recognize codons on the mRNA by being complementary in the base pairing, like we have seen that between the coding and template strand of the DNA. And so here, this is a part of the tRNA molecule that will read and recognize the information in the messenger RNA molecule. And here on the opposite end, we have the acceptor end of the tRNA molecule. This is where the amino acid is attached for, or the amino acid is attached that is encoded by the matching codon for the anticodon here on the tRNA molecule. And then, of course, we need the machinery to bring messenger RNA, tRNA, and with tRNA, of course, the amino acids together, and this happens in cell organelles known as ribosomes. And here we see what happens in such a ribosome. So here the ribosome runs down the messenger RNA strand and finds the matching tRNA for one of the codons on the messenger RNA, and if that matching tRNA is found, it will attach the encoded amino acid to the already existing amino acid chain. So in the end, we get that long chain of amino acids that was previously encoded in the nucleotide sequence of the messenger RNA, and of course with that also in the nucleotide sequence of the coding strand of the DNA. So this is basically what happens during gene expression, where we go from the gene on the DNA, on the coding strand, on the DNA encoded in the nucleotide sequence via messenger RNA to the protein, where that information was used to assemble amino acids in the required sequence to synthesize the protein that is encoded in the DNA. So what we have as a result of gene expression now is our protein that we need in hopefully the correct amino acid sequence, so that the result is a working copy of that protein. Could be an enzyme, could be an ion channel, could be a transporter that is then transported to the cell where it's required to do the job. An ion channel will be embedded into the cell membrane. A structural protein will be joined with other proteins of the cytoskeleton. So this is how cells work and put gene expression to work to produce the proteins that are required to do the job.